So,
It's not that.
no, it's not. So we'll do two different examples of meiosis, I suppose. So we'll have a cell that's got, we'll start with the same cells. And this is basically replicating what we talked in class yesterday, but again, maybe if I do this a little bit more explicitly than we did in class, it will help solve any conceptual issues. So here are two pre-synthesis diploid cells, the red chromosomes and the blue chromosomes. So what are these going to look like after synthesis? How many copies of red are there going to be?
It's going to be 2 copies. 2 copies.
For each of those, so each of those lines doubles, right? So we go from 4 copies of DNA to 8. And the sister chromatids are held together, just putting a little dot in between them. So we know those are, each pair of sister chromatids are the exactly genetically identical versions. The 2 copies you got from your mom versus 2 copies you got from your dad. What about the one on the right? Does it look the same or different?
It'll be the same.
Be the same. So up until this point, we're just replicating DNA. Meiosis in 2 different cells in the same individual is happening in the same way. So now I need to put in some annotation. So these are the mom and the dad versions of the red chromosome, and the mom and the dad versions of the blue chromosome. All right. So, time for meiosis I. What's going to happen?
You're going to get
So let's say we're going to have, let's say that cell's going to divide along that axis. The same thing is going to happen over here.
You're going to have one of each on each side, but they're paired up.
So yeah, so which, we're going to arrange these, each of the chromosomes forms a tetrad. So the 4 versions of red all bundled together, and the 4 versions of the blue chromosome grouped together. So that one dyad, which is one pair of sister chromatids, the ones with the dots in between them, one dyad's on each side of the cleavage plane, the cleavage furrow where the cell is going to divide. Cleavage plane, or cleavage furrow, where the cells divide. That's cell biology, you don't need to know that for this class, but where the cell is going to start dividing, the cleavage plane. It's where the cell cleaves. So here's your question, if I understood correctly. So where are we going to put the M's and the D's in the bottom half of this picture? We know that it's going to be the two sister chromatids going together. So we could put the two M's here. That's always going to be the case. So the two sister chromatids are always going to go together into the same daughter cell at meiosis I.
So it's mom, dad, dad.
In this case,
Yeah.
has to be dad, dad on the bottom then.
Yeah.
So what do we do on the blue chromosomes?
It could also be mom, dad, dad, or you can flip it.
Right. So I'll do the first one you said.
Okay.
And that's because these chromosomes are totally independent molecules. They line up however they line up at metaphase during the cell cycle. So it's not the case that because the mom's versions are on top on the left side that it has to be that the mom's alleles of the chromosomes are on the right side as well, on the top. Now, that's in one cell that's undergoing meiosis. Now here's meiosis happening in a totally different cell, in the same individual, in the same gonad, different cell.
We still did mom, dad, dad for the red one. It could be dad, dad, mom.
Right. So, and that's just two meioses that we've looked at in this one individual. How many meioses does this individual do in, let's see, it's a human, male. How many sperm does a human male produce over his lifetime? I have no idea, but
Lots.
it's lots. So, like millions, maybe? So on average,
The combinations can be as much as or as different as the different alleles that are there, that are available from the first synthesis.
right, so we've got the maximum of 2 alleles per individual, and we're looking at 2 genes, or 2 chromosomes in this case, the red chromosome and the blue chromosome. So if you look over millions and millions and millions of meioses, how many times will this orientation that's on the left happen? By chance, just randomly. If you do this 1,000,000 times, how many times will the orientation that we drew here happen?
25% or 50%.
There's only 2 options. We drew them both. So it's 50%. So half of the time, we'll get this random arrangement with mom's alleles going together with mom's alleles. The other half of the time, it'll be mom's allele going with dad's allele. So if we zoom back in, we can finish the first division. We've got the alleles that were inherited from mom going together into gametes, and the same is true for the alleles that came from the dad. Same picture as above, just after division happens. And if we go over here, we can finish off this drawing. Where we have the other possible combination. So now what happens during the second division? That's just the first division. So take each of those cells. We're going to produce the 4 meiotic products, and we're going to do the same thing on the other side. We're going to see what we get. every one of these gametes has to have
Right.
one copy of each chromosome that we're talking about. In this case, we're only drawing 2. Of course, in humans, if I wanted to be really explicit and show you exactly how it works, I'd have to draw 23 chromosomes. My tablet's not big enough to do that. I can't zoom in far enough to draw 23, so I'm only showing 2. Hopefully 2 is enough to make the point. Okay. So right now, all of these gametes are identical. They're haploid, they've got one version of each chromosome. It's which version of each chromosome that's different in these, on the left side and on the right side.
Well, these first two would only, they all have the moms. Because that's all that's available.
Right?
And the other ones would only have the dads.
Right? And those, by the way, are called parental types. For future reference. That's not important for this exam, but we'll hear this term again.
Because they don't have one of each, is that what it is?
They're called parental types because these look like the gametes that were used to create the individual who's going through meiosis.
Oh.
So this is kind of like what
Like
their
in the
mom's
beginning.
this is like what their mom's oocyte looked like that was fertilized to produce this individual we're talking about. And on the right side here, the one with where chromosomes all came from dad, or have dad's version of the chromosomes, those represent the genotype that was present in the sperm that dad contributed to this individual.
And over here, you'd have one mom, one dad.
Right, so on the left side, as I drew it, the mom would be the red chromosomes, dad would be the blue chromosomes. Dad's allele, that is, dad's version. And then on the right side, it would be dad is red and mom is blue. And those, again, for future reference, are not surprisingly called non-parental types, which is also a term that is related to one we use more frequently, which is their recombinant types. They have new combinations. These gametes have new combinations of chromosomes.
This is how they create like new strains of
This is how genetic variation comes to be.
in biochem we were talking about recombinant DNA, and they can like.
So this is related to recombinant DNA, but this is natural recombination.
Oh,
This isn't.
like.
This is recombination that happens in meiosis in individuals just based on sexual reproduction. The recombinant DNA that you talk about in class and we'll talk about in the future is based on actual in the laboratory manipulation of DNA to make new combinations.
Then if I understand this process,
Okay.
so when you say apply it to.
So how would we apply this? It's all about Punnett squares, and it's all about application in genetics. When we're talking about transmission genetics, is often setting up crosses and predicting the outcomes of crosses. So the important point to take from what we've done so far is to realize that we have 4 different types of gametes. We've got the parental type with mom's alleles. We've got the parental type with dad's alleles. We've got the non-parental type where red is mom and blue is dad's, and we've got the non-parental type where red is dad's allele and blue is mom's allele. So really, there are 1, 2, 3, 4 combinations of alleles that can be made by one individual in all of their meioses. So regardless of how many of their cells go through meiosis, there's only 4 possible haplotypes of gametes. These are the 4. and they will occur in this ratio. There are 2 number 1s produced, 2 number 2s produced, 2 number 3s produced, and 2 of the 4th type. So it's a 1 to 1 to 1 to 1 ratio. Not when you look at an individual meiosis. So you look at one cell going through meiosis, you're only going to see 2 types, right? The left half of the screen or the right half of the screen. But when we talk about, we're setting up a cross between plant A and plant B, there's an assumption in there, which is that we're talking about, assume you're talking about all of the possible sperm or eggs, pollen or oocytes, that are produced by that plant.
So this is where we would put the information into a Punnett square.
Exactly.
The first one you really only need, since they're homozygous.
Right. So what were the 4 types? So let's do this. Here's a Punnett square, and this is the Punnett square you would draw for a dihybrid. You've got chromosome, let's say, so we're going to still do red. So red is mom, or blue, mom's chromosome. And we've got then dads. So there was a red dad with a red mom, there was a red dad chromosome with a blue mom chromosome. So those are the 4 different combinations. Go ahead.
There's 2 moms?
Well, I'm sorry. That should be... I'm glad you're paying attention. I did that on purpose. No. There. So we got all 4 possible combinations of 2 versions of 2 genes.
This one, you mean at the end?
So this is after meiosis. It's exactly like what we finished on the previous slide. So we get down to the bottom. There are 4 different types of gametes that are produced. These are the 4. So if you took an individual that had let me again be explicit and draw the genotype of that individual. So 2 blue chromosomes, 2 red chromosomes. This was the diploid cell. So this Punnett square we're drawing is what happens if you crossed 2 of those individuals together. Or selfed a plant. It was like this. So both of these individuals have exactly the same somatic diploid genotypes. After meiosis, either one of those cells would produce that set of gametes. 25% would be red M with red D, and so forth. And so we would have to fill that in down here too. So we have, this is where the independent assortment, by the way, comes in. So M and D, separate chromosomes, M and D are different versions of the same chromosomes. Red is one chromosome, blue is another. So the red M can go with red D, or it can go with, has to be, right, blue D. And the blue M can go with red D, or the blue M could go with blue D. And those are, again, those are the 4 types. They're always present in the exact same frequencies. 1 to 1 to 1 to 1. So if we set up this cross, if you fill in this table, which I can't because it's too small, if we zoom in there,
you really only need the 2.
we get 2 M's, one from one parent, one from the other, 2 red M's, and 2 red D's. And if we scroll over, then we'd fill in the next one, which is 2 red M's. And this time we've got a red D and a blue D. And so you could do that and fill in that whole table. And that would tell you the genotypes, all of the possible genotypes that would exist if you crossed those two individuals together to produce the next generation.
But in the Punnett square, it doesn't, well, maybe when we use the uppercase letters and the lowercase letters, then you can tell which one's the recessive and which one's the dominant genes. And
right. So you can, yeah, if you have information, you can tell something about dominance. So yes, we're not talking about dominance yet, but we could.
Eventually that will play a part.
Yeah, that's important as well. So
right now we're just looking at all the possibilities.
Right. So let's throw in some dominance though. Let's say that on the red chromosome, mom's allele is dominant, so I'm going to use that black apostrophe on mom's allele to demonstrate that it's dominant. And let's say on the blue chromosome, dad's allele is dominant. And let's say the M prime allele makes you have curly hair. I'm just making stuff up. And let's say the D prime allele gives you blue eyes. Maybe I should have said red hair. That'll be easier to remember, right?
That would help us now when we're looking at.
So what are the?
So we have to put in where the primes go, right?
Let's see.
On the outside.
Well, yeah. Don't worry about the primes. That's my own nomenclature I'm using just for this. That's not
yeah, I know.
standard genetics.
But I'm saying on our Punnett square, we're going to have to write out which M is the prime. There's only one
right.
red M is prime, not both.
That's right. Yeah, you're right. You're right. So
Either one.
it's the red version of mom's chromosome 1. So we don't have to have this nomenclature.
Oh.
This is the red M that's dominant.
Oh, okay.
I've just added another layer. By adding this mark, I'm doubly coding what's dominant. So yeah, we could do this though. It doesn't hurt.
And then the blue D is
And the
dominant.
blue Ds are dominant. Okay. So what are the phenotypes of the two parents, these two cells over here on the right?
So one has red. They have both the red hair and the blue eyes.
That's right. I erased those, but I should put those back.
They both have red hair and blue eyes. So
Right. So what we've done is we've taken a red-haired, blue-eyed individual and mated it with another red-haired, blue-eyed individual.
So more than likely, it's almost 100% that they're going to come out with red hair, blue eyes. Almost.
Well, that's a great question. So what's the probability that we get how many kids of theirs? That's exactly what the application of this meiosis dihybrid cross Punnett square is. What are their kids going to look like? And what's the probability of getting, so let's do that question first. What's the probability of getting red-haired, blue-eyed kids? At least 50%. I don't know. Let's work it out. So the first thing to do,
Because unless they have both recessive genes, then it wouldn't show up.
so let's do it the easy way. Let's fill in the upper left quadrant. Let's actually fill in all of the genotypes. So do you agree with that?
Yes.
Okay. So then, if we add these little tick marks,
You don't have to.
which we don't have to, but for consistency I'll do. All the blue D's and all the red M's are dominant. Okay. So what are the different phenotypes that we have here? So
Well,
3 of these.
that first one has only red hair, no blue eyes.
Red hair, that's it. We don't know what the recessive phenotype is. We didn't say what eye color you have if it's not blue. It's not blue.
Not blue.
Well, probably not blue. Okay. For the other 3, they're the same.
2 or
They're different genetically.
but because D is the blue D is the dominant gene, then it would make that they all have blue eyes and red hair.
In other words, yes, absolutely. In other words, every one of those squares representing a genetic individual, a type of individual, a genotype, has a tick mark for each of the 2 chromosomes.
Or like when we did the worm things.
So those are all what genotype, or what phenotype rather.
Blue eyes, red hair.
Yep. Blue eyes, red hair. Because every one of those individuals has at least one dominant allele. Now we could finish filling out the Punnett square, but I'm not sure that that's actually necessary.
It's gonna look the same in each of the other 4 sections. So the other 3 sections are gonna look like.
Where else on this table will you find a square that definitely has the red M together with the blue D?
Red M with the blue D. Here? Wait, let's see.
So,
Red M, blue D.
so the red M is only going to be in the top 2 rows, and the blue D is only going to be in the last column.
Or,
Well, we've filled out this column.
but here too then, or no?
So we've got D primes going all across here.
All here they don't have.
Right.
the-
Here we don't have the right D, so we're not going to get dominant phenotypes for eye color in that
yes,
row. And in this column, we're not going to get
Eye color either.
eye color.
Or red
And in neither of these
or red hair.
are we going to get red hair or in the 3rd row or in the 4th row for hair color.
Okay.
So what fraction of individuals have both dominant phenotypes? You predicted 50% earlier when you said, I think, half, or it's going to look the same in every quadrant. What really turns out being the case.
It's only those 3.
It's right.
Yeah.
Well, it's all 4 of those individuals. So this whole upper quadrant. Oh, so you're right. Those 3 have the primes. So we've only got 3 out of 16 individuals that have both dominant traits. So we started with 2 individuals that have red hair and blue eyes. We mated them together, but only 3/16, because there are 16 squares here. So there are 3 out of 16 theoretical kids. That would be the frequency. If these individuals mated and had an infinite number of kids, 3/16 of them would look like the parents. The other 9/16 have some other combination of traits.
Oh, they might have red hair, they might have blue eyes, but not both.
Right.
Or neither.
Right. Yeah, so you get red hair, blue eyes, you get red hair, normal eyes, you get normal hair,
Red
normal,
hair.
red hair, normal eyes, blue eyes, normal hair, and then you get some normal eyes, normal hair. I shouldn't say normal when I'm talking about hair color, but
Or eye color.
or eye color for that matter. Exactly. Then what you would have to do if you wanted to do the whole Punnett square is you would fill in every square. You would look at the genotype in that square, figure out its phenotype just based on what's dominant and what's recessive. And then you could total up. This is not going to be on the exam, but you could then total up for those 16 squares, what are the 16 different phenotypes? What are the 16 different genotypes? And what winds up happening, don't freak out, is you wind up getting a genotype ratio that's that. 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1.
We talked about it.
So this is in the chapter. So there are so that is 16 different 16 different individuals in the ratio of 9 different. You get 9 different genotypes in this table. And when you look at them in combination, there's a 9 to 3 to 3 to 1 ratio of phenotypes. So there are 4 different phenotypes represented in this table in those 9 different genotypes. And we already talked about what the 4 different phenotypes are. It's the 4 different combinations of eye colors and hair colors.
And we thought, oh, okay, well then for sure, but if the recessive genes are still there, some of them no.
Here's another question that I could ask related to a Punnett square like this, which is related to the rolling, glowing worm questions. Oftentimes, scientists who are interested in crossing different individuals together to make a certain combination of traits want to be able to make a lot of the type of individual that has the trait that they're interested in, like rolling, glowing worms, for example. So in that case, what you're often looking for is you want to generate a pure breeding individual for those traits. So there are lots of ways to make worms that glow and roll, but they're not necessarily going to produce 100% of their offspring that have the exact same traits. Just like we saw here. We started with two parents that look phenotypically identical, same hair color, same eye color, but because neither of these parents is pure breeding, when you cross them together, you get...
So if it is pure breeding, then it would for sure.
Right. So one question I might ask related to this Punnett square is, if we crossed these two individuals that have red hair and blue eyes, what fraction of their offspring that have red hair and blue eyes that look like the parents will be pure breeding for that trait?
So just one. Just one, then the one in that bottom one. Right, because they've got both dominant genes on for the red hair and the blue eyes.
Exactly. So you had 3 red hair, blue-eyed kids, and one of those 3 is homozygous. That means they're pure breeding. So if you took that one individual and you selfed it, him or her, well,
then you would get another purebred.
Then its gametes, what is carried in the gametes produced by this individual?
It's just those. There's the recessive ones are not.
Has to be a big M, red M with a blue D, because they're homozygous. There's only one type of haplotype that is in the gametes that individual produces, and if you self it, you're making combinations of gametes that have red M's and blue D's with gametes that have red M's and blue D's, and you make 100% offspring that look exactly like the parent. So in this case, we've got 1 out of 3 individuals that look phenotypically the same will actually will be pure breeding. The other 2, if you selfed this individual or selfed this individual, sometimes you would get other phenotypes out of that selfing cross. Now this individual is pure breeding,
For the recessive genes.
but in this case it's for the dominant version of, what was that? Red M?
Red hair.
What was red hair? So this is a red-haired normal-eyed individual pure breeding for that trait, but not the one we were looking for.
Okay.
So you could look at the whole table and find every square where there's pure breeding or not, but for all of the pure breeding cells in here, they're not necessarily for the right combination of traits that we were seeking in this case.
Okay.
So there are other pure breeding genotypes in here that we haven't filled in